Good day. Good day, everyone. Good morning, good afternoon, good evening, wherever you are, and welcome to the this IUIS Immunopedia Frontiers webinar for today. We have an exciting talk today on the Ebola virus infection from Dr. Anita McElroy. She is a pediatric infectious disease physician with a research interest in hemorrhagic fever viruses. She holds an appointment as assistant professor in the School of Medicine at the University of Pittsburgh. She is a member of the University of Pittsburgh Center for Vaccine Research and provides clinical care at UPMC Children's Hospital in Pittsburgh itself. Her lab focuses on the mechanisms by which hemorrhagic fever viruses causes disease, and I think we're going to hear quite a bit of that today. Anita's webinar is sponsored by Fluidine. Thank you very much. And I would also like to request that you place your questions in the Q&A section of the platform and not in the chat box. And what I will do as moderator is collect these questions and moderate the discussion after Anita's talk. My last thing is please check out the IUIS website for future webinars in the next coming weeks. So thank you very much, and over to Anita.
Thanks, Clive. I'm super excited to be here with you guys today and looking forward to telling you about our studies into Ebola virus pathogenesis. Thank you so much for the opportunity to share some of our data with you today. And I'm going to talk to you about how we went about mapping human immune responses to Ebola virus infection using some somewhat novel technologies. As a disclosure, I just need to let you know that I'm receiving an honorarium for this webinar. I'm going to start by telling you a little bit about Ebola and Ebola virus disease. I assume if you've logged into the webinar, you have a little bit of interest in this area, so this is going to be quite brief. And then I'm going to give you an introduction to the CyTOF technology, which is what we use to conduct these studies. I'll describe the cellular immune landscape that occurs following Ebola virus infection, and I'll compare and contrast the CyTOF data with data that has been obtained from other studies performed by both our group and others. And then finally, I'll correlate the immune findings with key features of Ebola virus disease and share with you some novel findings that we discovered using these methods, and then demonstrate that they are repeated with unsupervised analysis. And I'll go into more details as we get there. So let's start with a little bit of information about Ebola virus. It's a filovirus. This is a negative stranded RNA virus that gets its name from its filamentous structure. It is generally accepted to have a bat reservoir, although that has not yet been conclusively proven. It does cause very severe and often fatal disease in humans, with case fatality rates ranging from 30% to 90%, depending upon the strain that causes the infection. So this is, of course, one of the world's most deadly viruses. It is transmitted person to person via bodily fluids or through blood, and it is found in Africa, in sub-Saharan Africa, and in West Africa. And you will all, I assume, remember the large West African outbreak that occurred in 2014 over a several-year period where this region of the continent was affected. There's a lot of information that was learned in the context of that outbreak. We now have vaccines that have been developed. We have therapeutics. The field has rapidly advanced because of what happened during that time frame. I'm going to show you data from humans. And when humans present with clinical disease, they tell you when they first had symptoms. So all the data is reported day post-symptom onset. And that is somewhat subjective because as I note on this slide, Ebola virus has a 2 to 21-day incubation period. So the time between actual exposure to the virus and development of symptoms will vary from individual to individual. So just keep that in mind as we go through the data. Ebola virus disease itself is characterized by fever and fatigue and weakness, myalgias, diarrhea, and nausea. And of course, these are all very non-specific symptoms. These are much more likely to occur in someone in these regions of the world who have malaria or typhoid fever. So the clinicians who work in areas where Ebola is prevalent have to have a really high degree of suspicion to think about this disease as the diagnosis. When patients experience severe disease, it manifests in a manner similar to sepsis. So these patients have coagulopathy, shock. They can have encephalopathy and respiratory failure, but they end up with multi-system organ failure, and that's usually what is associated with their demise. On the right-hand side of the slide here, I'm showing you data from a study we did in Sudan in several years back. The outbreak was in 2000 and 2001. And we demonstrate that people who have high viral loads are more likely to succumb to their disease than people who survive. And this data has been repeated over and over again by multiple groups, especially coming out of the West African outbreak. So a high viral load is a true harbinger of a bad outcome for Ebola virus disease. The study cohort that I'm going to take you through today is a study cohort that only exists because of what happened in 2014. So while the outbreak was ongoing in West Africa, many people traveled from all over the world to assist, to provide clinical care, logistical assistance, diagnostic support. And while individuals were there, some of them contracted Ebola virus disease. And all in all, 27 people were repatriated back to the US or to Europe for clinical care. 4 of those individuals ended up at Emory University in Atlanta, Georgia, where I was working at the time. I show you some snapshots from the news. These were pretty stressful times. The community was not very excited about patients with Ebola coming to Atlanta, Georgia. But all of these individuals received care at Emory University Hospital's Serious Communicable Diseases Unit. And the fact that all 4 of these individuals survived their disease and that there were no secondary cases is a real testament to the quality of the infection prevention, infection control, and medical care that was provided by the clinicians in that unit. The CDC happens to be about a mile away from the Emory University Hospital. And so the samples were all handled in the BSL-4 containment lab at the CDC, which is, of course, the kind of laboratory conditions in which Ebola needs to be handled. Fortunately, all 4 of these patients really graciously consented to participate in collaborative research efforts using their residual clinical specimens. You will see that I will show you data today that is very longitudinal, many years out for some of these patients. And that was samples that were collected specifically for research. However, all the data from the acute phase of their illness was done using residual specimens. So very small clinical samples were taken as part of their clinical care, and then residual samples from the acute phase were used for research. So that's one limitation to the work that we were able to do because we had very little specimen to work with. So this is the first bit of data that we gathered from the context of this outbreak. And we published this in 2014 or 2015. Showing that in the context of human Ebola virus disease, there's substantial immune activation. And so you're going to see slides like this a couple different times throughout the talk. And each time you will note the name, the code of the individual. So EVD 2, 5, 9, and 15 were the 4 patients. And if you read any of the literature that we published on these patients, the nomenclature is, of course, all the same throughout, so you can directly compare the data from study to study. You'll see on the x-axis down here the day post-symptom onset. Keep in mind this is somewhat subjective. This is self-reported by the individual. And you'll appreciate that for some of these patients, for EVD2 and EVD5, they didn't come to clinical care until a week or 2 into their illness. Whereas for EVD9 and 15, they actually were admitted to the unit much earlier on in their disease course. EVD9 becomes quite important because EVD9 is a patient for whom we had multiple samples and who was admitted for clinical care even prior to the time at which their viral load peaked, right? And then we had samples from them well into their convalescence. I'll take you to the table here on the right because it's important to note that there were some clinical differences between these patients. So EVD 2, 5, and 15, we've categorized as having moderate disease. And we do so based upon the fact that they didn't require any extracorporeal support. They all had elevated LFTs. You can appreciate the alanine aspartate aminotransferase levels were relatively high in all of these individuals. They all had thrombocytopenia, meaning they all had low platelets. But EVD9, not only did EVD9 have excessively elevated liver function tests and thrombocytopenia, but EVD9 required ventilation, dialysis, and was in fact encephalopathic. So this is an individual who, in the context of typical Ebola virus disease cared for in rural settings and endemic areas, would not have survived their disease. This person is alive because of the supportive care that they received. So this is an example of a very severe case, and we'll utilize that information as we talk about their immune responses. On these graphs, the blue dots just represent normal, healthy individuals. So you and I, we don't have any plasmablasts or activated CD4 or CD8 T cells in our blood normally. And plasmablasts represent activated B cells that are actively producing antibodies. And so you can appreciate that all of these patients have much higher than normal levels of activated B cells, activated CD4, and activated CD8 T cells. And then you can correlate that with their viral loads. And as we go through each cell type, I'll talk to you in a little bit more detail about that data. So because we had such limited sample to work with, we realized this was a pretty unique cohort of cryopreserved PBMCs from individuals with Ebola virus disease. This is not the kind of sample that is easy to come by. And I happened to be at a meeting at NIH on Ebola, and I met Gary Nolan, and he gave a talk there about CyTOF technologies. And he was talking about using their experience using these large multi-parameter flow panels to map out human immune responses, both in health and in disease. And so he and I talked and decided that this would be the perfect way to utilize these precious cryopreserved PBMCs to look at Ebola immune responses over time in the subset of patients. And so their panel that they have developed includes both surface and intracellular markers, as well as several phosphospecific antibodies. And I show you here another example of the viral loads in each of these patients, and I'm showing you the time points that I selected for the CyTOF analysis. Again, limited by sample availability. So we tried to select a sample that was from the most acute or earliest available time points for each patient. So you'll see an acute phase time point for all of the patients. And then you'll also see a later time point at a time when they were either convalescing or had control of their viral load, or at least in the case of EBD2, evidence of viral load control, like the viral load was declining. As I mentioned before, we had a lot more samples for EBD9, so we used 4 different samples from this particular patient, and we were able to use a sample from the most acute phase, a sample from the time point at which their viral load had peaked, a sample when they had viral load control, and then a sample even further out into convalescence. So this is the sample set that you'll be seeing today. Let me just give you a little bit of intro into the CyTOF technology so that you have some familiarity with it, just in case you don't already. So instead of using fluorophores, CyTOF uses isotopes. And as this periodic table demonstrates, there's lots of different options, even within a particular element. And so each one of these bars represents an atomic mass of a particular isotope that can be coupled to an antibody to use in mass cytometry. The beauty of the atomic mass spectrum versus using fluorophores is a lot of the detection capability has similar intensity. So the detector sees one tag in a similar way that it sees another tag. Whereas with fluorophores, you might have a very strong BB421, but maybe BB786 is much weaker. The other beauty of using mass cytometry is because you're not looking at a spectrum of fluorescence, each isotope has a unique mass. You don't have that signal overlap, and you don't have to compensate like you do with traditional flow cytometry. And then the last real advantage to mass cytometry is you don't have to deal with background from autofluorescence that you might see, say, in macrophages that would spill into your FITC channel. So these are the real strengths of using mass cytometry. The instrument that we used in our study is called a CyTOF2. I show you a picture of it here, and I'm just going to walk you through how this works. So cells are generated in suspension. They are labeled with antibodies that are coupled to these particular differential mass isotopes. And the sample is then aerosolized and nebulized, and such that you have a single cell per droplet. And that is then ionized, and the whole sample is essentially destroyed in the process. So all the cells, all the biological material is destroyed. But what is left are these specific isotopes. And these isotopes are then selected and separated out by something called TOF, which is time of flight. So there's a direct correlation between the mass of the isotope and the time it takes to travel from the input to the detector. And so using that distance, the system can categorize each particular isotope and understand the quantities and quantitate the amount of that isotope on that particular cell. And then the system integrates that data and generates what we're more familiar with looking in a traditional flow plot, where you can look at the amount of a particular marker on one cell versus the amount of a different marker. And this gets really complicated really quickly. So when you look at the panel that we use, right, over 40 markers, the gating strategy can be quite difficult. So those of you who are used to looking at flow cytometry, this looks quite familiar. I just want to walk you through how we did some of the initial gating, and then we'll talk about specific cell types. So the ability, you have the ability to do live dead staining, and so we did that. So we're only looking at live cells when we do these analyses. And I'll just remind you that, again, I processed all of these samples from residual specimens, but they didn't sit. Every time a sample was taken, it was processed within 8 hours. So this is really high quality PBMC preps that we're starting with. You can also gate out doublets, so you can take your live cells that are singlets and you can eliminate, in this case, RBCs. You'll note that there are not a lot of RBCs in this preparation because it's a PBMC preparation. So by definition, you've already purified away the RBCs. We also can gate out neutrophils, which you'll see here. There's very few neutrophils, again, in a PBMC preparation. And that gives us our leukocytes. So these are CD45 positive cells that are not RBCs, that are not neutrophils, right? That are all live cells. And this is the basis from which we do all of the additional gating and analysis. And I will show you different data throughout the talk where we're comparing back to the frequency of a particular cell type relative to the total leukocyte population. And when I say that, I mean all the CD45 positive cells. So in the interest of time, we can't obviously talk about all of this data, but what I want to do is focus in on these particular cell populations, and I'll take you through the more details as we go. So we're going to talk about monocytes. We'll talk about B cells, which includes both plasmablasts that are not class switched as well as ones that are class switched. We'll talk about CD4 and CD8 T cells. I'm going to show you some data on really unique cell populations that we would have never known to look for if we hadn't done such a multi-parameter analysis. And then I'll show you some really novel data on myeloid and plasmacytoid dendritic cells. And then finally, just demonstrate to you a little bit of work on unsupervised clustering from this data set. So let's start with monocytes. So monocytes have been associated with loss during acute EBD and with death from EBD. So you'll see down here on the bottom, anytime I reference anyone's work, I'll show the title of the paper and the authors. And this work is from Anya Ludka in Cesar Munoz Fontella's lab. This is a group out of Hamburg where they did work in Guinea during the West African outbreak. They used residual samples from patients and they assessed the monocyte profile in these patients. And they were able to see that of the 3 different types of monocytes, either CD14, which are considered classical monocytes, CD16, which are considered non-classical monocytes, or CD14, CD16 double positive monocytes, which are considered intermediate monocytes. In all 3 cases, patients who had Ebola virus disease, shown in red here, had lower levels of these cells than patients who had other febrile illnesses. That was their control group. In addition, they were able to see a relationship between these non-classical CD16 positive monocytes and clinical outcomes. So the patients who died here in green had lower levels of these cells than the patients who survived. Now, this study done by Anya and her colleagues used samples that were obtained approximately 4 days post-symptom onset from all of these groups. So we're really looking at the context of acute disease, which is one of the benefits of our CyTOF data because we could do longitudinal analysis in individual patients. And so I'm showing you the monocyte data for that patient EBD9. You're going to see plots like this several times. Anytime I show you this data, I'm going to show you a control. So on the far right, you're looking at the same cell populations that you might see in a normal healthy control. In EBD9, we saw almost a complete loss of all of the monocyte populations here at the very earliest time points. And our day 6 would be equivalent to sort of the data that I showed you from the previous study. So clearly monocytes are lost during acute infection. And Anya also demonstrated in her paper that monocyte loss correlated with viral load. So the more virus you have, the less monocytes you have. But what we learned from this CyTOF data was that there's recovery of those monocytes. So here at day 14, you really start to see them coming back. As you go to day 20, you start to see the intermediate cells coming back. And by the time the patient is in convalescence, you really see a decent, almost normal spread of all of the different monocyte populations. What was really fascinating was because we had some intracellular markers included in these panels in the bottom right, we were able to show that the monocytes at day 14 that were CD14, the ones that show up here, they're actually proliferating. They're actively proliferating because they're Ki67 positive. So the idea is that this population comes in to basically reproduce all of the cells that were lost over time. So it's pretty well established that Ebola infects mononuclear phagocytic cells, both in vivo and in vitro. But we don't know if this loss that we see in the acute phase is because the cells are being destroyed by direct viral infection. That's entirely possible. Or if perhaps those cells might be lost in the blood because they're trafficking to sites of inflammation. So we know especially the CD16 positive population, so these non-classical monocytes, they're also considered patrolling monocytes, and they're known to traffic to sites of inflammation. So one of the reasons that these are lost may be because there's a lot of inflammation in the tissue. This is the challenge that we always have when we're doing studies in humans, right? We can only sample the blood and we can't sample the tissues. That is being overcome by some investigators, but certainly not something we could do in the context of Ebola virus disease. So let's talk next about B-cell maturation. So this is data from a paper published by Carl Davis in Rafi Ahmed's lab using samples from these same 4 patients who were cared for at Emory. And these folks, like I said, agreed to participate in longitudinal research. So the x-axis here is different from the x-axis that I've shown you before. We are now 900 days post-symptom onset. So these patients were followed for years. And in the top left panel here, what I'm showing you is their class switching over time. And so the human heavy chain locus is organized such that IgG3 is first, and then IgG1, IgG2, and IgG4. So as antibodies become more class switched, they become less IgG3, and you can appreciate this decline in these patients, and they become more IgG1, 2, and 4. And we don't see a lot of IgG2 in these patients, but you definitely see an increase and you see IgG1, and then as time goes on, you see more class switching to IgG4. And what's really striking is EBD5 kind of stands out from amongst the rest with the most IgG4 class switched antibodies and still pretty high level antibodies. If I take you down to the bottom left panel, when Carl looked at these 4 patients, so the color coding is switched a little now, each color is a separate patient. Carl looked at these memory B cells, that's what MBC stands for, and he assessed the frequency of these memory B cells in each patient that bound to the glycoprotein, the Ebola virus glycoprotein. So these are virus-specific memory B cells, and EBD5 again stands out as having the most for the longest time. In addition, he assessed the avidity of the circulating antibodies in these patients, and EBD5 again stands out as having the most antibodies for the longest time. And he looked at somatic hypermutation. This is a measure of how much affinity those antibodies have. So they get more and more mature, they mutate, and you'll see EBD5 again over time has the most affinity maturation of their antibodies. So EBD5 is really unique in this cohort. But when we looked at EBD5 using our traditional flow cytometry methods, and we measured plasmablasts as a function of total CD19 positive B cells, we really didn't see EBD5 looking all that different from the other patients. However, when we did our CyTOF analysis, we were able to see a couple of unique things. And so now I'm showing you data from all 4 patients, and you'll see this sort of layout a couple of times. Again, day post-symptom onset is always on the x-axis. The y-axis here is now percent of leukocytes. So remember I told you when we looked at the gating strategy that the data I was going to show you was going to be all as a function of total CD45 leukocytes. So the numbers are going to be smaller than you saw in the previous graphs when we're looking as a function of total B cells. And so you can appreciate here that all of the patients have time points at which they have higher levels of both non-class switched and class switched plasmablasts, but EBD5 clearly stands out as an outlier and a pretty unique patient who has much higher levels of these class switched and non-class switched plasmablasts. So clearly plasmablasts are elevated in acute infection. This particular patient stands out both in our CyTOF analysis and in the analysis that was done by Carl Davis. But we were also able to look at some of the intracellular signaling molecules in these cells because these were included in the panel developed by the Nolan lab. And you can appreciate here in the upper right that naive B cells, all of them except from EBD15, express phosphostat1, and that the class-switched plasmablasts also express phosphostat5. What's really cool about the phosphostat5 data is Dave McElroy, who's been working in the Nolan lab for many years, has recently done a study showing that H1N1 challenged humans have plasmablast expansion, and that those plasmablasts in the acute phase of the infection also express phosphostat5. So this probably represents a fundamental feature of the human immune response to viral infections. So you'll see a little bit more of Dr. McElroy's work later in the talk. Antibodies are really interesting in the context of Ebola virus disease. There's some old data from outbreaks that occurred many, many years ago, demonstrating that some folks who succumb to disease do so without ever developing any antibodies. But there's never been any large cohort studies directly comparing antibody quantity, antibody function, antibody quality in patients who live or die. So this is an area that I think really requires a lot more work. And there's clearly some heterogeneity to responses from individual to individual. The last thing I want to say about B cells is, of course, B cells make antibodies, and we know that that antibodies are important, but we really know that antibodies are important in Ebola, because one of the things that came out of the West African outbreak was the development of monoclonal antibody therapeutics. And so 2 separate monoclonal antibody therapeutics, MAB114, as well as the Regeneron polyclonal, it's a mixed product. Both of those have been shown to have efficacy in survival and increasing survival. So in patients who were really high viral load with a 90% mortality rate, that mortality rate was brought down to 60% using monoclonal antibody therapeutics. So B cells in Ebola virus disease are going to be really important. So let's move on to T cells. So this is the data that I've shown you before from our initial studies, right? These very limited studies that we did using traditional flow cytometry. And you can see that all of these patients have activated CD4 and activated CD8 T cells. And we defined these as a function of total CD4 or total CD8 T cells, and activated markers that we used was HLA-DR and CD38. These are pretty commonly used in the field. And this is all that was known about T cell activation in Ebola until Paula Rubial did this study in Cesar Munoz-Fontela's lab. This is again work they did using residual samples from patients in Guinea, where they looked in a little more detail at these T cells. So CD8 and CD4 T cells are shown on the graphs. You can appreciate that they've got lots of activated T cells, whether they're using CD38 HLA-DR as the marker or CD38K67. But what's really important from their study that, of course, we weren't able to do in our small cohort is they're able to look at patients who live and patients who die, and they don't see any difference in the level of activation between those 2 patients. But when they looked at CTLA4 and PD1 markers on those activated T cells, they saw that patients who died had higher levels of these markers. Now, these markers are known to increase in the context of acute expansion, and there's a direct association with a lot of viral load and patient outcomes being negative. So it may be that these markers are upregulated because there's a lot of viremia in these patients. But it's also possible, because these markers are known to have negative effects, regulatory effects on T cells, that these are sometimes considered markers of exhaustion. So it's also possible that these patients have T cells that aren't really effective. And Dr. Munoz-Fontela's group has actually gone on to demonstrate that there really is an ineffective oligoclonal T cell response in fatal cases, whereas in contrast, patients who survive, they have a real nice polyclonal breadth, right? He did this by sequencing the T cell receptors and showed that they have a nice breadth of reactivity against EVD, against Ebola virus. So there really is data suggesting that T cells are activated in any patient that gets Ebola virus disease, but they're really effective in the patients who survive. So something is making an inhibitory response on those T cells in the context of severe disease. And I'll have some hypotheses for that in a minute. When we looked at the CyTOF data to look at our T cell responses, again, we're looking at the percent of total leukocytes. We see activated CD4 T cells like we would expect in all 4 patients, but really something striking showed up when we looked at activated CD8 T cells. And you can appreciate this patient, EBD9, who I'll remind you had very severe disease and would have died without supportive care, has essentially no activated T cells in the context of total leukocytes, which means there's a lot of cells that are present in this sample that we're somehow missing. So we went back to our data and we asked, what are the majority of the leukocytes in this patient that we're somehow not characterizing by our traditional gating strategy? And here's what the CyTOF data taught us. So now we're looking at the top at lineage negative, so CD3 and CD19 negative, CD14 and 16 negative. So these are not B cells, they're not T cells, they're not monocytes. And we look at HLA-DR staining. If we look at the HLA-DR negative cells, this is what I want you to focus in on. You can see that there's, at day 6, at this early time point, EBD9 had pretty similar frequencies of these populations as the control patient. But by day 14, 20, and 28, you see a massive expansion in this HLA-DR negative population. So we went to this population of cells and we looked at their expression of other markers. And so on the bottom panel here, I'm showing you not a lot in day 6, but day 14, 20, and 28, right? Massive expansion of these particular cells, and they turn out to be CD11b positive. And when we looked at some of the intracellular stains on these CD11b positive cells, we found that they're phosphostat3 and Ki67 positive. And in fact, these cells, these lineage negative 11b stat3 Ki67 positive cells, make up 17% of all of the leukocytes on day 14 in this patient. So this is a massive expansion of this cell population. We went to the literature to try to understand what these cells could be, and they seem to look like cells that are present in the blood during a process called emergency hematopoiesis. So B cells and T cells, they recognize antigens, and they proliferate in response to recognizing antigens. But myeloid cells are not antigen specific. So what happens is they get stimulated to be recruited from the bone marrow by expression of growth factors. And this population of excess myeloid cells have sometimes been called in the literature myeloid-derived suppressor cells because they're seen in sepsis and other critically ill patients, very similar to patients with Ebola virus disease. These cells produce IL-10 and TGF-beta and are thought to contribute to immunosuppression, which might be an explanation for why we see immunosuppression in such severe EVD. And we even have some data to suggest that that's the case. So these are two other studies that we did. On the left, you're looking at a kinetic analysis of biomarkers in a cohort of patients, but I'll just draw your attention to EBV9 because he's the patient I'm showing you data for. High levels of IL-10 in this patient in the acute phase, consistent with immunosuppressive activity. High levels of MCSF, consistent with stimulation of the bone marrow to push out a lot of these myeloid-derived cells. And we even saw elevated levels of MCSF in patients who succumb to their disease in our old study from biomarker data looking at patients from Sudan virus outbreak. So there's pretty clear evidence that there might be emergency hematopoiesis in severe Ebola virus disease. So let's move on to the last cell population that I want to cover, and that's dendritic cells. And so I'm just showing you here, again, data from EVD9 from those 4 time points. The control is on the far right. We're looking at myeloid dendritic cells as defined by CD11C expression and plasmacytoid dendritic cells as defined by CD123 expression. You can appreciate that the myeloid dendritic cells essentially disappear in the acute phase, and then they return in the convalescent phase. We see a little decline in Pdcs during the acute phase, but it's not nearly as dramatic as you see in for the myeloid dendritic cells. And so we looked at these myeloid dendritic cells in a little bit more detail. And we went back to Anya and Cesar's paper where they looked at patients from Gabon. And in their supplement, they look at CD1C positive dendritic cells, and they see that they are lower in patients with Ebola virus disease than other acute illnesses. So we looked at CD1C on our myeloid dendritic cells. So as I mentioned, they disappear in the beginning, but as the myeloid dendritic cells come back at day 28 here, this is the pink, or at 19 at the second time point, you're looking at data from all 4 of these patients, you can really appreciate that there's a striking upregulation in the level of CD1C on these myeloid dendritic cells as they return in these patients. Now, CD1C is an HLA MHC class 1-like molecule that is important for presenting lipids. So this may play a role in adaptive immunity in Ebola virus disease, but that is certainly something that isn't quite well understood at this point. Pdcs, I think, are the other area where this study really taught us a lot about Ebola virus disease. So plasmacytoid dendritic cells are responsible for the vast majority of the stimulation of the adaptive immune response, presenting antigen to B cells and T cells, and they also make a lot of interferon alpha. They're responsible for the majority of interferon alpha production. And we knew a little bit about these Pdcs in the context of Ebola infection because data from the Bassler lab in 2 separate papers has shown us that Pdcs are not infectable by Ebola in vitro, which is kind of unusual because every other myeloid cell is infectable. And Pdcs are also not susceptible to VP35 inhibition of interferon. And so VP35 is a protein made by Ebola that's known to inhibit interferon, and it does so in every other context except in Pdcs. So the sort of conclusion from this data was that perhaps Pdcs didn't really play a role in Ebola. And I would suggest to you that our data are considering that might be not the correct interpretation. We really think that Pdcs do play a role in Ebola because in vivo, in these patients, when we look at Pdcs, we see upregulation of CD38 at these acute phase time points. So this is going to be the marker in blue in all 4 of these patients. And this upregulation of CD38 has been examined in other studies of humans in response to viral infection. So I mentioned earlier, I would show you some additional data from David McElwain's group. And what you can see here, this is in individuals that were given H1N1 challenge. They were given live flu virus, and their PDCs upregulate CD38 again in this acute phase, same time frame that we're seeing with our patients. And you can appreciate the histograms here on the right where you see this massive shift, CD38 upregulation at these early time points. But what is really striking about David's data is that he's able to demonstrate that if you block CD38 signaling over here on the right, you inhibit the ability of these PDCs to make interferon alpha. So this CD38 presence on these PDCs is consistent with the fact that CD8 signaling is occurring, and we know that it's required for interferon alpha production. So there may be a really important role for PDCs in the context of Ebola virus disease. The last bit of data that I want to share with you are some lessons that we learned from unsupervised clustering. So when you do these multi-parameter analyses, it's kind of difficult sometimes to sort through all the data when you're just doing traditional gating. So folks turn to other methodologies. We used scaffold plots. And so what we've done here is I'm just showing you a comparison of 2 time points for each patient. For EVD9, remember we had 4 time points, but I chose the time points that were pretty similar to those that we had for the other patients for this comparison. So we're looking at day 14 versus day 28 for EVD9, and then the first and second time point for each of the other patients. These samples, in order to do this analysis, the data is downsampled to the minimum number of events in any one sample, and that's completely random. And then you ask the program to generate clusters of similar cells and to show you where they lie in space in relationship to one another. And we asked it to generate as many as 200 clusters. And so when you do this kind of analysis, you get a plot that looks like the ones I'm showing you right here, where you can see that similar types of cells group together. So the monocytes are together with the other myeloid cells. You see the B cells off on their own, and K cells are off on their own, and then T cells are off on their own. What I want you to appreciate from this particular graph or depiction is that we're showing you the fold change in cell abundance. That's what the heat map is indicating on these particular clusters. And you'll see a lot of blue on here. So what that means is at these acute phase time points, a lot of the different immune cell types present in these patients was declining in abundance, or was lower in abundance than at later time points. We've done some Ki-67 analysis in the same kind of format, and you can also appreciate that the same cell populations that are low in the beginning, they are proliferating and they end up coming up later at later time points, like I've shown you throughout some of the talk. But the things that I found most interesting about this particular clustering analysis was not the cells that were low, because we can see that pretty easily, but there's cell populations that are high, and I'll bring your attention to cluster 41 here. So all 4 of our patients have increased levels of this particular cluster at the acute phase of infection, right? The first versus the later time points. And when you look at this cell population, because when you have one of these clusters, you can have the program show you essentially all of the histograms of all of the markers. And this is a lineage negative CD11b, Ki-67 positive, phospho-STAT1 positive population. So back to this emergency hematopoiesis, myeloid-derived cells that are present in the blood of these patients at these early time points. And obviously there was a lot more present in EVD9 who was more ill, but we still detected them in all 4 of the patients. Okay, so what have I shown you today? I have shown you that monocytes and myeloid dendritic cells decline in the blood during acute Ebola virus disease. We don't really understand if they're being destroyed by direct viral infection or if they're being recruited to sites of inflammation. I've shown you that T cells are activated, but they aren't always effective. And the scenarios in which they are not effective are the patients that don't do well, because polyclonal responses are associated with survival. That's data from Cesar's group. B cells get activated, but their quality differs significantly in various hosts. I think there's a lot more that can be done with B cells, and this is an area that is ripe for future study. Pdcs are present and activated during Ebola virus disease, and I would suggest that this means they are probably contributing in an important way to adaptive immunity, and that we need to understand this a little better. And then finally, in severe cases, emergency hematopoiesis is activated, and this could be the reason why we see immune suppression in patients who have severe and fatal EBD. And this suggests that this is a really great novel area for potential therapeutic intervention. So I just want to end by acknowledging the people who did the work. So everybody who's underlined on this slide in the Nolan lab, at Emory, and at CDC, all directly participated in work involving samples from these patients. There's a number of different funding agencies that have assisted with this work. And then also on this slide are some of my current funders and my current lab, which is located in the University of Pittsburgh, if you're interested in checking this out. And I thank you for your time and attention, and I'm happy to take any questions.
Great. Thank you very much, Anita. Hi, there's a number of questions in the Q&A box, which I'm sure you can read, but I'll read them out anyway. From Wendy Gilmore, very nice work, agree. I know you include MSD, monocyte-derived suppressor cells, but did you include any other markers of regulatory monocytes or B cells or NK cells, Treg cells, for example?
Yeah, so FOXP3 was included in our panel. If you're interested in looking at exactly what we did in our published paper, it's in JCI, and the supplement is a listing of all of the different markers that were used in that panel. And FOXP3 was included, but there was no real signal that stood out to us for T regulatory cells. I didn't look in detail at anything else. One other thing about this study that I think is really important is, you know, we're doing kind of a cursory look, but there's a lot of you out there who are really experts in specific cell types and specific cell populations. So I'm happy to make all of this data available. All you have to do is email me, and I can send you the raw FCS files so that you can kind of drill down on a particular cell population if you're interested in understanding them. That was a really important aspect of this work. We wanted to make the data accessible by anyone. So feel free to email me. You can find me, Google, easy. At my PIT address, and I can give you access to the FCS files if you want to look at a particular population.
Yeah, that's great. That will be really great for some people to nail down that population. I guess on a similar vein to that is the Treg cells. Did you have Th17 markers there in your panel?
No, IL-17 was not included in the intracellular markers that we had. Most of the intracellular markers were focused in on phosphorylation proteins, and that's because the Nolan lab had developed this panel specifically to look at some of the phosphorylation signaling molecules intracellularly as part of their panel. I do have to say there's one caveat. So when the Nolan lab developed that panel, they specifically, when they collected whole blood samples to use this panel on, included phosphatase inhibitors in their preparation. So we didn't collect these samples with that in mind. So our samples don't have the phosphatase inhibitors included. So during the 8 hours or so, you know, maximal that it might have taken from the time the sample was drawn to the time I was processing, there would have been potential activity going on. So you have to take the phosphorylation data just a little bit with a grain of salt because we have that limitation in our data set.
Yeah. Another question about the heterogeneity of the B and the T cells. How is that related or associated with the viremia or the viral load of the patients? Was there some association there?
Yeah, it's a little bit hard to say with a sample set of 4 only, but we certainly see higher T cell activation associated with higher viral load, right? And that was also seen in Cesar's study as well when he looked at patients in Guinea. So the more active, the more viral load you have, the more antigen stimulation, the higher levels of activated CD4 and CD8 B cells as a function of total CD4, CD8 T cells. So that's been seen in several different studies. You know, the other caveat I have to share about this particular patient data set is some of these individuals received experimental therapeutics. So they were initially cared for in 2014 at the very beginning of the outbreak. You know, there was a lot going on. We didn't know hardly anything, honestly, about how to care for patients with Ebola in the, you know, Western critical care setting. So a lot was learned. And so some of them received experimental antibody therapeutics. Some of them received experimental siRNA therapeutics. And those are listed in that first PNAS paper. If anyone wants the details on that, I'm happy to talk about that more. So the immune responses that we observe in these patients need to be considered in the context of the fact that they also received some therapies that could have affected their immune responses.
Right. I think that's an important caveat, yes. I guess there were only 4 patients, but I guess when you have longitudinal data, you want to look at rates of decay or rates of change. I mean, it's difficult to draw conclusions from only 4, but did you look at any of that, especially for the antibody responses?
Yeah, so that would be Carl Davis's work, as he was looking at really truly longitudinal data over years. There certainly are declines in all of the patients over time. I don't think a specific rate of decay was calculated, but I think that's probably feasible. If you were interested, you could reach out to Carl Davis directly, since he did most of those B cell studies. Regarding some of the cellular populations that we looked at, it was quite interesting. So EBD2 and EBD5 both received a ZMapp, so a monoclonal antibody therapeutic. And they both had sort of prolonged T cell activation, longer than we saw in some of the other patients, right? Out to 2 months post infection, we were still able to detect a lot of activated T cells. And we hypothesized that that might be because the monoclonal antibody therapeutic was able to pick up antigen, and those antigen antibody complexes were picked up by dendritic cells or some other antigen presenting cell, and maybe resulted in sort of prolonged antigen stimulation of T cells. And that might have been why we saw really in those 2 patients who received monoclonal antibody therapeutics, prolonged activation. But again, N of 2, so it's just a hypothesis.
Sure. A very specific question. Why does VP35 not inhibit type 1 interferon secretion by plasma dendritic cells? What is the mechanism? What is special about these cells in terms of RIG1 detection? Simply that do they not get infected? Question mark.
Yeah, these are fantastic questions. So it's all out of Chris Basler's lab. He's in Georgia State in Atlanta. His group is the one who has done that work. So they were the ones who demonstrated that if you take whole blood from humans, normal healthy people, and you purify myeloid cells or monocytes or Pdcs specifically, so they're already, Pdcs by CD123 staining, those just didn't get infected in vitro, which was a pretty unique finding, right? Like, why would they not get infected? And that VP35, which functions by inhibiting RIG-I, just that's like I think you're alluding to, doesn't seem to be active in those cells. And I don't understand why it's not active in those cells. But there's been also some data published that Chris Basler's been a part of. So if you remove VP35 from Ebola, right, it no longer inhibits interferon and is not pathogenic anymore. It is essentially a vaccine in a non-human primate. So VP35 interaction with RIG-I and its inhibition is really, really important in pathogenesis for this virus, and why in the context of PDCs, they don't get infected, or it doesn't seem to be active, I think is an important thing that deserves some additional work.
Mm-hmm. Has this been extended to maybe the field, or getting field samples from West Africa? Obviously you can't do this unless there's another outbreak, which of course we don't want. But yes. How would you go about addressing that issue?
Yeah, so a lot of the work that was done with larger cohorts was done by the European consortium. So I mentioned some of Cesar Munoz-Fontella's data, folks out of the UK like Miles Carroll. There's some colleagues out of Italy who have also done some of this kind of work. Most of these samples were all obtained from residual specimens in the context of clinical care. So to my knowledge, no one has done any true research-based, research-driven studies over time in patients other than the ones that we looked at here. There's been a lot of convalescent-based research work done, right? Looking at responses over time in patients who are survivors, but not necessarily in this sort of acute time frame. I understand from Cesar Munoz-Fontella that WHO has an interest in trying to set something up so this kind of work can be done prospectively, because unfortunately there will be another outbreak at some point, right? We sort of know that. And if it can't be rapidly contained with ring vaccination, which has worked really effectively, it sounds, in the last few outbreaks, or with the use of monoclonal antibodies as therapeutics in patients who get disease, then there might be a role for additional research, but that hasn't been formally established to my knowledge.
There's a couple of questions on a very similar theme of HLA associations. Again, N=4 is not, but has there been a broader association with HLA and responses to Ebola?
Yeah, so there's some old data from a long time ago. There's some KIR, so natural killer cell receptor associations with outcome. And there's a little bit of HLA data from a Sudan outbreak. This was from many, many years ago. I don't know of any recently published HLA data from the West African outbreak, but I have every expectation that that's something that people are looking at in more detail. We did HLA type all 4 of our patients that were cared for, but with an N of 4, you can't really say a lot. We did that HLA typing specifically so that we could develop tetramers in order to evaluate their virus-specific responses. And so in our initial PNAS paper, we have those tetramers, and we did do some tetramer-specific staining, and we've also done ex vivo peptide stems to try and understand which are the antigen targets of the T cells in the context of infection.
So that actually is the basic basis of the next question is, what are those antigens targeted by T cells?
Okay. So we see a lot of nucleoprotein that gets targeted. That seems to be, it's the most highly expressed protein by the virus. So it makes sense that if there's a lot of that antigen around, it's going to be targeted and identified and targeted by T cells. But we also saw a lot of glycoprotein specific activity as well. A lot of the other proteins, we didn't look at the polymerase, but we did look at all the structural proteins. They didn't seem to have a lot of activity. So the nucleoprotein is the most highly targeted, followed by the glycoprotein.
Interesting next question, which is around Ebola and COVID-19 and the cytokine storm that is most likely in both, but how do they differ? Is there any? We obviously have a lot of data on COVID-19, but
yeah. I mean, I think when you look at very severe viral infections in general, you tend to see some very common phenomenon in patients who have very severe disease. COVID-19 seems to be quite similar as well. So patients with severe Lassa disease, patients with Crimean Congo hemorrhagic fever virus disease, patients with Ebola, patients with severe COVID. In all of these scenarios, even certainly dengue, this has been looked at a lot. Common themes include a lot of IL-10, a lot of IP-10, a lot of MCPs, and a lot of IL-6. So I would say the same kinds of cytokine perturbations that have been seen in other severe viral infections are the ones that we see in Ebola. So I think these are probably common fundamental features of sepsis and or severe viral infection. So there's quite a lot of similarities.
Other question, your opinion in vaccination of classic vaccine. I'm not quite sure I understand that question, but maybe I can reframe it in terms of, I mean, there are, you mentioned the ring vaccination strategies, which have been proven to be very successful. What is the vaccine response? How does that differ from the natural infection? I mean, you get a good T cell response with the vaccines.
Yeah. So.
And the natural
the Ebola virus vaccine. Sorry, the Ebola. We had a little delay there. Technology.
Yeah.
COVID times. So the Ebola vaccine that is currently being used, that's been licensed, is a vesicular stomatitis virus vectored vaccine, and it expresses the Zaire strain glycoprotein. And so this means that the immune response to the vaccine is really only targeted towards the glycoprotein. So it's not actually a vaccine response that's targeted towards any other viral proteins. And in the context of viral infection, of course, you have a breadth of a response. One of the real markers of vaccine efficacy, and these studies have largely just been done in non-human primates to date, right? That's where we have most of our data. That particular vaccine seems to be really important on antibody responses. So it seems to be a B cell mediated protection, whereas other Ebola vaccine platforms, so there's some chimpanzee adenovirus platforms and there's some other adenovirus vector platforms, seem to be more dependent upon the T cell response to provide protection. So I think the vector that you use, like the type of vaccine that is used to generate the immune response, really changes the mode by which it provides efficacy. So I don't know that has been conclusively shown in humans, whereas it really has been kind of clearly defined in non-human primates.
Right. a question that I also had myself, which somebody's also asked, is your NK cell populations. I mean, they were there, but how many? I mean, we know that there can be many populations of NK cells. Which did you see a narrowing or an expansion of these populations? Did you?
Yeah. We didn't have time to talk about the NK cell data. So we did look at CD56 and CD16 on NK cells. We defined them by CD7 positive. So these were the cell populations that we examined. We did see Ki67 positive NK cells. So they are proliferating in the context of acute disease. We see some early declines. And that's actually consistent with some data from an Italian group where they do look at NK cells in residual samples from patients from West Africa. And they see activation of certain cell populations. We did not use exactly the same markers on NK cells as they did in their study, so the data aren't directly comparable. But if you look at our JCI paper, there is a little bit of data on NK cells, and we have a couple of NK cell markers. So if you have a specific interest in that population, you can take a look at it.
Great, thanks. So another comment and question from somebody. Hi, Anita, great talk and fantastic work. We are seeing a lot of data emerging for emergency hematopoiesis in severe COVID-19 cases, disease, and the immature neutrophils are correlating with increased disease severity. Any thoughts on this in relation to your EBD9 patient?
Yeah, that's an interesting observation. I really wish I could tell you about neutrophils. Unfortunately, I prepped PBMCs, so I lost them all, right? Just by definition of the way I prepared the samples. Right. We did not look at neutrophils. And even in our initial studies where we used whole blood, I didn't do any specific staining or gating for neutrophils. In the very beginning, I was using a 4-color Accuray cytometer in the BSL-4. So you can imagine with 4 colors, you can't really do a lot. So we didn't look at neutrophils in detail.
Yeah, and I guess a related question might be, would a potential lymphoid lineage emergency hematopoiesis, for example, DCs and NK cells, be related to severe outcomes? Would you get this rapid expansion or constriction, I guess? Is that related to disease?
Yeah, the only data, I mean, we don't have cellular immunology data on that. The only kind of ancillary data we have is when we see elevated levels of MCSF, so monocyte colony stimulating factor, we just biomarker, we can measure it in the plasma, and we see that associated with severe and fatal disease. And we also see high levels of IL-10 putatively produced by these myeloid cells. I can't prove that either. So it's just ancillary data to suggest that this is an association. We don't have cellular immunology data.
There is another question here, which I'm not sure I quite understand it, but I'll read it anyway. In your opinion, in vaccination of classic vaccines such as Ebola, BCG, polio, and now with the COVID mutations, I guess the variations, and mucormycosis, I'm not sure what that means, but yeah. Sorry, Randa, you asked this many times, but I'm not sure what that means. So we're almost finished the session. Keep your questions coming. We have 2 more minutes. I noted that your phosphostat 5 expression in B cells seems to be associated with recovery. But now we know that, well, I found it intriguing that in the 2014 outbreak, there was about a 50% mortality rate, roughly. So that you've obviously looked in those that have recovered, which is great. What I mean, I'm asking you to conjecture or to hypothesize. What would you think is happening in those that don't recover? Which would you place your bet on as the protective immunity?
Yeah, that's a great question. I think we don't know enough about B cells in the context of acute disease. Like I sort of alluded to this in the talk. There's a little bit of data from outbreaks from many years ago. And there's small cohorts, 7 to 10 patients in outbreaks that occurred that demonstrate that oftentimes people who die of Ebola virus disease do so without ever having developed any virus-specific antibodies. So that really suggests to me that an acute and robust and early B cell response is probably going to be associated with better outcomes, but that has not been shown experimentally.
Yeah. I think on that note, we can end the session, and I want to thank you greatly, Anita, for a great talk today, and thank you very much. And please, everyone, stay safe, and also, while you're staying safe, look at the IUI's website and look at the future webinars. Thank you very much. Thanks, everyone. Good day.
Thanks, everyone, for joining from all over the world. It's great to see you.